Okay, so it's me again. Actually, one announcement I forgot to make, I want to make it just before I forget, and that is for in-person attendees, this badge that you have, you can use this to get past the security desk, tomorrow, Wednesday and Thursday, just, show this badge and you can kind of breeze through security. Sorry for not mentioning this earlier. Okay, so this next lecture is going to be, kind of simple. We're going to look at what is a cortical layer. And so the goals of this lecture will be just to review various definitions of what we mean by cerebral cortical layers, and also review some terminology for specific laminar features and types, to help make sure that you can navigate this literature. And also we're going to review laminar connectivity and the canonical microcircuit. Josine already mentioned this, and so I'll go over this briefly. These are, of course, important topics that anybody in the field needs to be familiar with. And then we'll talk a little bit more about blood vessel or so-called angio architecture and how it relates to the cerebral cortical layers. So again, kind of a simple lecture. So first off, there are layer structures in many brain regions, okay? But however, here in this course, we're really only considering the cerebrum and its cortex. And so cerebral cortex or cortex is Latin for bark. And so the name implies that this laminar organization of the cortex really is a defining feature. And so most of this bark is neocortex, which is evolutionarily the most recent. It comprises the isocortex, which has 6 main layer types with a rough common layout, as shown here. But we see across the cortex that there are differences, even within the isocortex. The other cortex, or allocortex, is about 5% of the remaining of the surface of the cerebral cortex. Again, fewer than 6 layers. We also have the paleocortex, which is older cortex. The archicortex is the oldest evolutionarily. It has 3 or 4 layers, includes structures that I'm sure you're aware of, such as the hippocampus. And there are other cortices sort of defined at the transitions between these different cortical types. Now, even though this basic 6-layered isocortex has a layout that's common across the cerebrum, we see that there are important regional differences as well, such as here you can see that, at least in this textbook depiction, the motor cortex appears to be missing its layer 4, although we heard from Josine that there appears to be a very thin, kind of hard to see if you don't look closely enough, layer 4 within this cortical region. But we see that there are regional differences, even within the isocortex, which provides the basis for defining cortical areas based on their laminar anatomy. So the laminar anatomy is really used to define cortical areas. Now, this groupings of neurons across cortical depths has been seen for as long as there have been depictions of the microanatomy of the cortex. This is a drawing from Ramon y Cajal. You could appreciate, in this drawing, a clear banding or layering of the neurons, across the cortex, across the cortical thickness. We see a grid-like structure of this anatomical organization, both in the radial direction as well as the tangential direction. Now, there are various anatomical definitions of layer types in the isocortex. They can be defined based on either cytoarchitectonic features like cell size or density, or myeloarchitectonic features such as myelin content and banding and fiber patterns. And so the isocortex exhibits these 6 common layer types, as shown here, and many sublayers as well, and also based on either cyto or myeloarchitectonic features, as you can see. And you can appreciate, while the basic 6 layers or 6-layer types are roughly present across the isocortex, I've heard it said that a 6-layered cortex is really a unicorn, in that the number of layers, the number of distinct layers that one can differentiate really varies a lot between areas. It varies a lot between different histological stains. And so it may be uncommon to come across a cortical area in the isocortex that has exactly 6 layers. And again, you can see there's various definitions of these layers we need to take into account. So I'd like to think of it as really 6-layer types that we see across the cerebral cortex. And so here's a drawing from an early edition of Gray's Anatomy, kind of showing the different definitions of the layers. So Farouk kindly labeled this in one of his recent articles, so I'm using that. Thanks, Farouk. And so Farouk pointed out here there's several names of these different layer types. Layer 2 is often called the external granular layer, layer 4 the internal granular layer, but really oftentimes we call layer 4 just simply the main sort of granular layer. All the layers above layer 4 are called supragranular layers. All of the layers below this granular layer are called infragranular layers. So it's called the granular layer because the cell bodies in this layer are kind of small, like grain-like granule cells like sand. And so we tend to see kind of very high density of neurons in this layer. And so as Jonthan said, typically if the cortex in humans is about 2.5 millimeters thick, our voxels these days for single shot EPI, I think we can achieve about a 0.8 millimeter isotropic without breaking too much of a sweat. You can see that generally we can only resolve maybe 3 kind of layer groups with MRI. We typically call these superficial layers, middle layers, and deep layers. And you'll see this terminology used a lot across both laminar fMRI and laminar MRI. And so again, we typically divide these into roughly superficial layers, which correspond to the supragranular, the middle layers, which correspond coarsely to the granular layer, and the deep layers, which correspond to the infragranular. So the other few features that I wanted to point out, we have both the external band of Baillargeat and the internal band of Baillargeat. These are heavily myelinated bands, both in layer 4 and part of layer 5. We see this a lot in the literature. Also want to point out this sort of fun layer at the top, this plexus of Exner. So layer 1, as Jonthan said, it's funny, my PhD supervisor used to call layer 1 kind of a copy of white matter. There's a lot of apical dendrites, there are some sparse, I guess, kind of GABAergic or kind of inhibitory cells there. Also some axonal terminations. So it seems that layer 1 has a role in terms of feedback and inhibition, but there's, very few cells there. Now, a very special feature is the stria or line of Gennari, which is a heavily myelinated band in layer 4B of V1. This is 4B, it's above the LGN input projections that terminate in layers 4C alpha and 4C beta. Here you can see the stria of Gennari in some of our old ex vivo data can be seen quite clearly. It provides a sort of anatomical definition of area V1, primary visual cortex, which is also known as the striate cortex because the stria of Gennari is a defining microanatomical feature. What's pretty cool is that even if you look at this fixed tissue, kind of raw tissue, you can see the stria of Gennari essentially with the naked eye. This might explain why it was first discovered in the 19th century before we had powerful microscopes. You can see the stria of Gennari pretty clearly in the raw tissue. Now, cortical area borders are often defined based on the cortical area's laminar features. We know that very famous parcellations, such as the cytoarchitectonic atlas of Brodmann, as well as various myeloarchitectonic atlases from Elliott Smith, but also Flechsig and the Votes, based on the different features across cortical layers, based on either cytoarchitectonic or myeloarchitectonic features. And so the cyto and myelo parcellations kind of roughly agree, but there are many differences. And I think that's still unclear what's the best way to define a cortical area. So we see here that, not every cortical area has a, purely distinct kind of laminar fingerprint. We see that there are roughly 5 cytoarchitectonic-based structural types of mesocortex, from agranular to granular. You see here that M1 is often referred to as an agranular cortex because it doesn't really have that granular layer 4, or at least it's quite subtle. We see that these primary sensory areas, such as primary somatosensory cortex S1, primary auditory cortex A1, primary visual cortex V1, tend to be granular cortices. They all have a really pronounced layer 4 in their cytoarchitectonic structure. Again, more groupings of these cortical layers, granular, agranular, dysgranular. We see this rough layout where the primary sensory areas tend to have granular cortices, whereas we see again here in motor cortex is labeled as being agranular. And I point this out not only to define terms, which is important, of course, but there have been proposals that there may be distinct functional roles of different cortex types, which leads to, I think, interesting hypotheses that the functional role of a given cortical area may be inferable by its laminar structural type. So I'm not going to say much about this. There's also subtypes based on myeloarchitectonics. We have unistriate cortex, bistriate, astriate, again, groupings of cortical areas, based on the myeloarchitectonic subtypes. So as Jonthan had also mentioned, we've seen that there's varying thickness of cortical layers across areas. Here again, we see that there's a really thin, often not really represented layer 4 in M1. And so what this tells us is that the layers are really shifting around across the cortex, even between adjacent cortical areas like V1 and V2. The thickest layers in humans are about 500 microns, as Jonthan said. And so what this tells us is that in a laminar fMRI, we very rarely, if ever, fully resolve the layers. We might need a 200 micron voxel in order to resolve these. What it also means is that, again, the cortical areas may have different expressions or different, biases towards different of these layers. So if I normalize the layer thicknesses by the full thickness of the cortex, we can see again how layer 4 in V1 is really huge compared to adjacent cortical area V2. And again, this may be a cause of sampling bias in fMRI. For example, in V1, since layer 4 is so pronounced, more of the signal may come from this cortical area. And so you may have heard different terms that we've used, either depth-dependent or layer-specific fMRI. You may also hear depth-resolved or cortical depth analysis. Even the Slack channel, I think, is called the depth-resolved fMRI. Why do we call it this? Why not laminar fMRI? Why the distinction between cortical depth and cortical layer? Well, this is important. It's because layer positions and thicknesses really vary across the folding pattern. We see here that the granular layer is sort of pushed up in the tops of the gyri. It's sort of pulled down in the bottoms of the sulci. We say that in sulci, the lower layers are expanded. In gyri, I'm sorry, I said it backwards. In sulci, the upper layers are expanded. In gyri, the lower layers are expanded. We see this from this beautiful data from von Economou. I think many of you know of the work by Bach, which also showed. So this change in the layer position really exaggerates the geometry of the granular layer, or layer 4. And what you can see is that there's sort of a non-trivial mapping between cortical depth and cortical layer. And so this is often addressed with the equivolume approach that Farouk will talk about in his lecture on layerification. But I just wanted to point out, this is an important distinction. We often infer the positions in the cortical layers based on the cortical geometry. It's hard to resolve the layers, and so many people, when they're trying to be very careful about their wording, they'll refer to a cortical depth analysis rather than a cortical layer analysis, just to acknowledge the fact that there's sort of, oftentimes we're kind of inferring or estimating which layer we're sampling from. So that's just a piece of terminology that's important to establish. We also see discontinuities in cortical layers between adjacent cortical areas. You can see this in the cell stain between striate cortex V1 or extra striate cortex outside of V1, such as V2. We see this sort of sharp discontinuity also in cell density and myelin stains. And so what this means is that this mapping between depth and layer is even more complicated, because not only does it vary across the geometry of the folding pattern, but it may vary between even neighboring cortical areas. So that's an important subtlety to keep in mind. Oh, I should point out that I think that these jumps are most often reported between kind of, around primary sensory areas. So this may be something that is not so prevalent in association cortex. So several layer features are detectable with MRI, luckily. We see in many contexts some contrast between these supragranular and infragranular layers, as shown here. We see differences in myelin content between infragranular and supragranular layers that can appear in MRI. We see, comparing to the histology on the left, the MR data shows us some contrast between supragranular and infragranular. This can be detected with a T1, as shown on the top, or T2 star weighted imaging, as shown at the bottom. It's actually pretty tricky to see such strong contrast in vivo. But the point is, there are, some features that can be detected with MRI. Here's another example. Layer features detected, such as the striae of Gennari. We see that, again, the striae of Gennari is defined as this heavily myelinated band of tissue in layer 4B. Often we see a lot of iron that co-located with the myelin. And so some combination of this myelin and iron gives rise to the strong R2 star contrast within the striae of Gennari. We see here that, the striae of Gennari, you can think of it as, again, a heavily myelinated feature. It's almost like a copy of the white matter, again, kind of sandwiched within the gray matter. So you can see that in this R2 star plot on the right, you see this sort of white band in the middle of the cortex, similar to the kind of the longer R2 stars seen in the white matter. And so, as Jonthan mentioned, there's distinct functional roles of different cortical layers as well as cortical sublayers, right? So we see here in this diagram, we see that there's different kind of ways to kind of both lump and split cortical layers. We see that layers 2 and layer 3 are kind of lumped together into functional layer 2, 3. We see that layer 4 is split. into layer 4A, layer 4B, layer 4C alpha, which is the magnocellular pathway involved in the wear pathway. We see layer 4C beta, which is part of the parvocellular pathway involved in the what pathway. And so we see here that there's different ways to kind of segregate, layers and split and lump them together based on their functional roles. Here's another example. Actually, maybe I saw this in your in your lecture as well. And so what's funny about this is that, do you guys notice anything that's missing from both of these plots? It's kind of funny, right? I mean, typically what we see is that, a lot of times when people are depicting the function of cortical layers, they don't even mention layer 1. Layer 1 isn't in either of these diagrams. Again, that's because layer 1 is very cell sparse. It's thought to have a limited functional role, although as Josine said, we see a lot of projections into layer 1, a lot of inhibitory cells there. So I do think it's quite important, but it's kind of common to not report any measurable neural activity within this layer. Now, perhaps the best known property of cortical layers is that specific layers are the sites of inputs and outputs along so-called feed-forward, feedback, and horizontal pathways. And many look to the seminal paper from Fellman and Van Essen for this general scheme. And so this was really kind of a meta-analysis paper based on demonstrations from prior work that forward connections can be distinguished from backward connections on the basis of the laminar organization of their terminations. And so they defined a so-called ascending or feed-forward pathway in which either the superficial layers or like a bilaminar origin projected into layer 4. They defined a lateral or horizontal pathway in which there is a sort of a, again, bilaminar input into sort of a columnar projection, as well as a descending or feedback pathway in which you tend to see an origin that's either infragranular or bilaminar projecting in a multilaminar way. And so this study came out around the same time, I think, as Douglas and Martin, that you also had mentioned, Josine, kind of describing a canonical microcircuit, which I think they defined based on electric recordings in the visual cortex in cats, maybe, I think. And so this canonical microcircuit, again, something that everyone here should be familiar with. It's an important topic in laminar fMRI. Sort of evolved a bit over the years. This is sort of a more recent depiction. Here again, we see, as Josine had mentioned, forward connections that project to layer 4, as well as backward connections, or feedback connections that project to layer 2, 3 in the supragranular cortex, or layer 5, 6 in the infragranular cortex. So a couple more details here. There's also appears to be some, recurrent connections, some inhibitory connections, both in, across all layers 2, 3, 4, and 5, 6. We also see intrinsic feed-forward connections, intrinsic feedback connections. But again, you can see why this sort of kind of 3 kind of compartment subdivision that we typically impose with fMRI, the superficial, middle, deep, is really pretty fortunate. It does map on to the feed-forward and feedback connections of the canonical microcircuit. This microcircuit has been observed in many cortical areas. Of course, does not extend to the whole cortex, but it's a pretty good starting point for any laminar fMRI experiment. Now, after defining these pathways, back to Fellman and Van Essen, they assigned each cortical area a place within a hierarchy. You can appreciate if cortical area is sending, a feed-forward projector into another cortical area, you may say it's kind of lower in the hierarchy. If it's receiving, connections from a different cortical area, you may say it's, higher in the hierarchy. And so with this logic, they computed a hierarchy of cortical areas based on the pathways identified between them. Now you can see here that this is a pretty useful schematic. Of course, it's a summary. There's many violations of this model, both functional and structural. And there have been some recent reappraisals of this kind of strict, kind of hierarchical view of the cortex. But still, we see that it's appealing to think of, if we can define these feed-forward and feedback projections as sort of, I think very attractive to try to assign a level of each cortical area to this hierarchy. Now, there are some subtleties to relate kind of laminar structural and functional connectivity. And so recall, Josine just told us about how neurons span multiple cortical layers. This is a kind of a famous picture from Henry Markram's shop, I think, showing how you can see here a bunch of neurons that are defined as being part of layer 6. They have these dendritic arborizations that kind of span all cortical areas, all cortical layers. And again, we see that, as Josie said, neurons are assigned a layer based on the location of the cell body. So what this means is that if we have some anatomical input projection into the superficial layers that, that synapses to the dendrites, say in layer 2, 3 here, when that pathway is engaged, where do we expect to see the neural activity? We see that it can be ambiguous. And so the point is to remember that neurons are not just, isolated soma, they do extend, and this can complicate the interpretation or the relationship between these known patterns of structural projections, to the functional connectivity. Now, it's another fact that we need to consider in laminar fMRI is that in many ways, the angioarchitecture of the cortex kind of reflects the functional architecture of the cortex. In fact, Duvernoy defined several vascular layers that kind of overlap with the cytoarchitectonic layers. Here we see, for example, this vascular layer 3, it's sort of a dense band of high capillary density that overlaps roughly with some of layers 3, 4, and 5. And this makes sense. We know that the vasculature is adapted to the needs of the cortex, and it makes sense that there'd be some relationship between vascular and neuronal architecture. Now, this vascular architecture also varies across cortical areas. This is my favorite example. Just to orient you, this is the calcarein sulcus in the human cortex and the white matter. We see that there's this dense band of capillaries within V1, and that sort of ends abruptly exactly at the V1, V2 border. Again, we see that a lot of the kind of microstructure of the cortex is recapitulated in the angioarchitecture. Here's another example. Again, in human cortex, we see that there's a dense capillary bed in S1 that ends abruptly at the M1, S1 boundary. So you can see this kind of close, relationship between angioarchitecture and functional architecture. And on the basis of this, Pfeiffer, who trained with Flechsig, actually proposed, we know about the parcellations of the brain based on cytoarchitectonics and Brodmann. We know about parcellations based on myloarchitectonics. He proposed a parcellation based on angioarchitectonics, which really differed quite a lot across cortical areas. This is a fun one. Farouk and I have been, marveling about this for years. So what he did here was, this is actually a photograph. He sort of painted these angioarchitectonic fields on a wooden model of the macaque brain and just photographed it. This is the lateral view. Here's the medial view. Again, defining angioarchitectonic field boundaries that roughly correspond to these cytoarchitectonic field boundaries. But what you can appreciate here is that there's a lot of complexity in the angioarchitecture or vascular architecture, which is really reflecting a lot of the functional architecture as well. Now, this is a course on the cerebral cortex, but I want to just point out briefly that these relationships can be seen in other parts of the brain as well. Here's a beautiful example looking at the angioarchitecture of the human thalamus. This is a vascular stain, again, by Duvernoy. So you see in this vascular density map that you can see like 6 layers of the lateral geniculate nucleus in the vascular density, which kind of mimic or recapitulate the 6 cell layers in the LGN. Just to point out that this kind of conspicuous relationship between vascular anatomy and functional anatomy can be seen in other brain regions as well. We see some variation of vascular density across areas. This is work from David Kleinfeld's group, looking at different types of cortices, agranular, partly granular, and granular cortex. We see that, first off, the depth profile, the cortical depth profile of cell density varies substantially across these areas. But he also saw that the depth profile of the vascular density also varied somewhat across these different cortical area types. And so we see that, less is known about the microvasculature, but we see that there's some kind of rough correlation in this case between vascular density and cell density with a correlation of about 0.64. Now, this is in mouse. I think other work in macaque, did something similar. Again, looking at the correlation between vessel density and cell density, pretty similar correlation coefficient actually in 0.64. But what's more interesting, and Josine also referred to this, there's an even stronger correlation between patterns of neuronal metabolism as inferred from cytochrome oxidase and vascular density, a correlation of 0.95. And so this makes sense. We know, again, that the vasculature is adapted, to the function of the brain. It's adapted to deliver energy exactly where it's needed. So it makes sense that regions that have higher metabolic needs would require more vasculature to deliver oxygen, to the neurons. And so again, we see that the brain's functional architecture is reflected in its vascular architecture. And I think this makes good sense. Okay. And so here again, some beautiful intercortical corrosion casks of the microvascular anatomy. We again see this sort of grid-like architecture of blood supply and drainage. I think this is human on the left, and macaque on the right. Josine warned me, yeah, sometimes these corrosion casks are tough, to generate, and so there may be some missing vessels in the human data. But again, you can appreciate the sort of grid-like structure that again is recapitulating, the neuronal architecture. As Josine mentioned, we know that there's some symmetry breaking in terms of blood supply and drainage in the cortex. We know that the blood kind of flows in from the oxygenated arterial blood is applied downwards. It flows in from the peal arteries, goes into the tissue from the surface. Most of the oxygen passes from the blood to the tissue in the capillary bed, and the deoxygenated venous blood is drained upwards from the tissue to the surface. So again, there's some asymmetries when looking at these patterns in the cortex. And as Josine mentioned, what this means is that when looking at BOLD, we're looking mostly at deoxygenated blood. Most of the deoxygenated blood kind of originates in the parenchyma and gets drained up to the surface. This is one of the reasons that we see these biases, more deoxygenated blood in these large draining veins in the surface. And so I'll wrap up by just posing this question, where are the control structures? So kind of a central question of laminar fMRI is whether blood flow is really regulated at a fine enough spatial scale to allow for distinct hemodynamic responses within layers, and whether this can be reliably seen across cortical areas. And so it's still not clear. As Josine mentioned, we used to think that the control structures with the smooth muscles that were aligning the kind of peel arteries, which would allow us to potentially deliver blood to individual cortical columns. But now we see that there are control structures along these penetrating arterioles as well, which could give rise to kind of a layer-specific, blood flow response. Will allow us to deliver blood to a specific cortical layer, so that we're not kind of, if the neuroactivity is in a particular cortical layer, this means that not the entire cortex is being flooded with blood. There's some evidence that there may be control structures even closer to the neural activity. Precapillary arterioles appear to have smooth muscles, and there's some intriguing evidence that there may be blood flow control at the capillary level. And I think what's interesting is that we see a lot of empirical data that suggests that there can be blood flow regulation at these fine spatial scales. It's sort of remarkable to me that it's still not really known what the ultimate kind of biological resolution of fMRI is, how fine, both in space and in time, is blood flow regulation in response to neural activity. It seems to be at the spatial scale that can allow us to see activity within 200 to 300 microns. We can discuss that more over the break. But I just want to point out that there is a limit to blood flow regulation. We just don't know where it is yet. And so this is a really important question to keep in mind when interpreting the data. Okay, so let me just wrap up. Main takeaway points from this lecture. I've shown you how the esocortex has 6 layer types, but not all areas have 6, proper layers. Depends on how you define them. This sort of 3-layer, I think some people call it like a super layer, which is a little bit confusing, but whatever you want to call it, this 3-compartment scheme captures many relevant aspects of function, as I showed you. The superficial, middle, kind of deep scheme that we use a lot in laminar fMRI. I've shown you how layers are defined based on the architecture, both cyto or myelo architecture, or on function. The basis for these definitions, this is the basis for the definitions of cortical areas. But keep in mind that they're somewhat subjective, and these definitions aren't always in perfect agreement. I've shown you how these layer positions actually shift across the folding pattern, which is why we sometimes say depth-dependent fMRI. This is a non-trivial relationship, which you'll hear a lot about more later in the course. But of course, I've showed you, as many of you I'm sure are aware, that the cortical layers are inputs and outputs of feed-forward, feedback, and horizontal pathways. Also, we see intercortical connections and computations as well that are layer-specific. But keep in mind that not all connections and not all pathways have been mapped out, especially not in the human cortex. And so I think this is a big, potential opening for laminar fMRI to try to understand these patterns in humans. But I've also showed you, of course, that vascular architecture reflects the laminar or functional architecture. I think this can be both good news and bad news. We can maybe learn about metabolism by studying vascular architecture, but it also may impose some biases in our fMRI data, and of course, challenges in the interpretation of our measurements. And so that's it. Again, kind of simple. I want to thank Renzo Furuksowski and also Josine for feedback. And I'm happy to take questions. I think we still have, yeah, we're actually doing pretty good on time. So I can take a question or two now, especially if anyone has questions.